3, go.
Hi, my name is Justin Zhu, and I'm a senior at Hamilton High School from Chandler, Arizona. Today I will be talking about a genomic sequencing tool I was able to develop at the Translational Genomics Research Institute. So basically, ever since the Human Genome Project was created, we are able to now successfully sequence a lot of the genes and identify mutations within the human DNA. So as a result, a lot of technologies have sprung up in recent decades that have allowed us to successfully identify new mutations that characterize cancer and other genetic diseases. One of the most prominent developments over the last few years was next-generation sequencing, which has allowed for these large quantities of genomics data to be successfully sequenced with greater resolution and allowing us to clarify these complex biological relationships. So as you can see, the process from going from chromosome to database has been faster and quicker than ever. So visualization in terms of genetic sequencing is important because it not only is able to help us condense information into these very clear models, but also because it allows us to be able to find variations and create greater and more accurate inference testing when it comes to identifying significant mutations within the human genome. So as you can tell, the process of looking at numbers becomes extremely easier when we're able to successfully graph these numbers into these pictures, allowing us to identify mutations successfully. So one of the programs that have been really prominently used by genomics researchers is the Integrative Genomics Viewer. The Integrative Genomics Viewer is created by the Broad Institute, and it's a program that allows us to group, sort, and filter DNA sequences from patients by having us modify the many features present in the IGV. Specifically, what we see here is that there's a console system that allows the user to interact visually with the DNA sample from the patient. And then within the program itself, it stores a lot of memory information. Kind of like when we're driving a car, what we see as the wheel is actually controlling these individual motors inside the car. So that's essentially how the IGV functions. There's a user interaction portion, and then there's the computer code inside that allows it to implement more complex algorithms. So in terms of interacting with the IGV, we're able to open up the application, and then in the left part of the feature, it stores the corresponding patient information, the DNA that corresponds to the patient sample. And then right next to it contains all of the mutations, variants, read counts that we want to look for to determine whether a specific cancer mutation, for example, is particularly significant for a specific patient. However, despite all these benefits that we see with the IGV, there are limitations in that mainly a lot of the features of the IGV are not automated. So, for example, when a researcher is trying to look for a specific variant that's occurring across a population of patients, they might have to kind of scan through and look through individual patients one by one. So as a result, the process becomes extremely tedious because as a result, much of the research data accumulation, data analysis has to be done by the researcher manually. So basically, in order to kind of identify these mutations within a shorter time span, we have to be able to find a method in which we can make the Integrative Genomics Viewer much more automated. So as a result, I came up with 3 goals I wanted to see accomplished with IGV, the Integrative Genomics Viewer itself. The 3 goals I wanted to see was customization, accommodation, and automation. Often when we're dealing with genomics data, a lot of the features that are available for researchers in terms of looking at specific mutations and how they want to view the mutations, there's not a lot of options, not a lot of flexibility for the researcher to be able to properly identify what he or she wants. So as a result, I want to be able to kind of integrate the integrative genomics sphere with a Java platform so that researchers with perhaps a more sophisticated understanding of computer science can have a more personalized approach towards analyzing their genomics data. Additionally, I want it to be accommodating because, as you may know, a lot of researchers have different ways of inputting their data. Some researchers might like putting decimals, some researchers might like putting fractions. As a result, IGV can't really accommodate all of these different syntax structures inputted by the researcher himself or herself. So as a result, if we can find a way to have IGV read in these different data structures, it's going to help increase the accessibility of IGV itself. And finally, we want it to be automated. This is probably the most important goal with my project because by having the IGV automatically search through the significant mutations without having the researcher look for all of the mutations in a given chromosome position, it will save tremendous time for the researcher and really help us encourage us to find significant findings to help patients better diagnose their DNA genetic problems. So as a result, this all comes down to increasing user-friendly with capabilities within the IGV. So the features I kind of came up with my program is that it's user-friendly, it's fully automated, and it's an interactive tool. It's able to eliminate, it's able to eliminate redundancies and limitations within the IGV. And also, in addition to all of that, it's able to create snapshots. So it's able to kind of store the information of the patient's DNA sample as pictures, and also read the significant mutations without having the researcher scroll through the entire DNA patient sample. And finally, output the DNA chromosome mutations containing the vital information. So this is kind of what my class structure looks like. It looks pretty simple. Essentially what we have is we're working with a main test class. It's the GUI, the graphical user interface that I designed. And within this graphical user interface, we're referencing several classes that help us accomplish various tasks that we need with the IGV. Specifically, the socket class allows us to connect with the IGV server, so we're able to directly implement customizable features of the IGV on our Java program. The file input class is able to read in all of those various syntax and file datas that the researcher wants us to analyze. The BAM VCF class is where we insert the patient's DNA information. All of the patient's information is stored within these BAM files and these VCF files. So that's going to be really helpful for us in being able to locate those mutations. And finally, we're going to be able to create a PDF report that contains all of those snapshots, those pictures of those patient mutations and the corresponding chromosome positions those mutations are located in for cancer patients. So as you can see, after several testing, my program was able to accommodate all of these different sample file inputs. So this first one I am showing you contains different columns of information corresponding to chromosome and the chromosome position for a specific patient. This next example shows an example in which chromosome and position are located in the same column. This is different from the first example because chromosome and position were located in different columns. And finally, the third example, we have some additional words and phrases inputted into our columns. For example, CHR1 instead of just 1. So as a result, the program is able to read through CHR1 as just chromosome 1, and likewise just the number 1 as chromosome 1 as well. So the procedures for operating my program is extremely simple. You open up my program, you specify the directory to save report, you choose your preferences, which range from the report format to to which BAM files to add, and finally you run the program. So now I'm going to actually show you my program so you know, I guess, what I'm talking about. So specifically I was able to kind of export my program as a runnable jar file. But basically, throughout this whole process, I've been working with my program on the Eclipse, which is a great platform for testing out my code and making sure that everything is running and debugging properly. So if you look closely, I guess, what we see here is that GUI class. So if we were to kind of scroll down the GUI class, we would be able to see that there's a lot of references towards these other classes, including a coordinate class, creating a PDF, and reading the PDFs, reading the file inputs as well. So to kind of operate this program, I will show you how to do it right now. So this is what my program looks like when you open it up. It also works by clicking on the jar file if you're using a Mac, or running with a batch script if you're using Windows. Essentially what you do first is you want to open up a new IGB session. So that's able to accomplish the goal right there by, as you can see, opening up the IGB directly from my GUI that I created. So now we have our IGB all set up. The server is connected to my program. So next what we want to do is we go back to towards our program, and we want to load our text files or our binary alignment maps, which contain the patient's DNA. So let's first add our patient's DNA. So as we navigate through this process, we will see that these BAM files contain a lot of, I guess, memory storage. So one of the key things I implemented in my program is that after 8 BAM files, it's going to cap it at that. Because if we add more BAM files, it's obviously going to take much longer for the program to run. And it's going to be relatively too much information to store. So I'm going to add a couple of BAM files right now. And one of the features I've also implemented was it only accepts BAM files when you're trying to add BAM files. So for example, here's a TDF file. If I press that, error bar pops up telling me I can't put that file down because it's not a BAM file. So essentially, yes, we add a text file. So I already created one here, so I'll just simply throw that in right here. And the text files I'm using for this example are comma script value text files. So everything is separated by a comma. What we do now is we can save it as under our test class, and we can proceed to run. As you can see, there's a lot of features we have. The genomes we are currently working with can include human genome, which is the default HG19, or the genomes of different dog samples, so like CanFam2, CanFam3, those correspond to the genomes of dogs. So now we run our program, and there's a confirmation box. I press OK, and we can proceed to go check it out in the IGB itself. So as you can see, it's locating through the chromosome locations and taking pictures of these chromosome pictures with the snapshots features implemented in my program. So essentially, after it's done navigating through the program itself, the GUI is able to close down, and we're left with a PDF report. So this is an example of a PDF report. The one I just created right now, as you can see, it's February 7th, 2017, so that was the one I just created. And as you can see, these are the snapshots of the different DNA samples for all of the chromosome positions. As you scroll down, you can tell that this sample doesn't really contain a significant mutation because not a lot of colored bars are showing up, indicating that perhaps this patient is not likely tested for cancer. So that's essentially how my program works. Right now it's being tested for a couple of cancer studies by researchers, by fellow colleagues I work with at the Translational Genomics Research Institute. So yeah, going back to my presentation, after we're done with running this program, we can see that it's able to accomplish the goals we set out. It's now automated for greater speed and less work done in evaluating the next generation sequencing data. It's customized because it's now easier to work with from a Java perspective. And finally, it's accommodated because all of those chromosome positions, those snapshots we took, were all automatically done by the computer. The researcher didn't have to scroll through manually. So now by combining the features of IGB and Java, we're able to now present a more optimized genomics tool for DNA sequencing. Finally, there are some limitations with this program. As I've indicated before, there are some discrepancies between the Mac and the Windows version. I'm currently kind of trying to find a way to incorporate both of them so that I'm not releasing two different versions for two different software systems. So that's something I'm eager to look forward to. And then future development, I'm hoping to kind of create this as an applet and then implementing some more features with perhaps SAM tools, which is another very versatile genomics tool that can help me characterize mutations with more accuracy and precision. And finally, kind of releasing this more as an application, not only for researchers, but perhaps for the general public as well, so people can kind of analyze their own DNA samples without needing to use, I guess, fancy equipment. So hopefully I can increase the accessibility of this product to a larger scale. And then finally, as I've mentioned before, the benefits of my project is that it's able to help specifically patients. That's our end goal with this entire project. We want to be able to identify mutations, identify diagnosis much more quicker with much more precision and accuracy for patients, especially patients with cancer. So as a result, by having this program being able to accommodate, being able to be customized and being able to be automated, it's going to be able to streamline the process for genomics research as a whole, and in turn allow for more quality treatment and quality care for patients. And I would really like to thank my mentors, Natalia and Dr. Kropp, for their mentorship throughout this entire process. I truly learned a lot more about the capabilities of Java and in terms of genomic sequencing. And finally, Translational Genomics Research Institute for providing me with the resources and the facility to conduct this project successfully. Thank you so much. Here are some references, and that concludes the end of my presentation. Thank you for listening.